get you started here. Thanks.
All right, well, thanks everybody for sticking around. Yeah, I'm Colin Callahan. I'm a graduate student in the Wildland Resources Department at Utah State, and I work in Karen Mock's lab. And I'm going to be talking about my thesis project and the results we have to this point. And I just want to say that we're dealing with a little bit of a different scale here, both spatially and temporally. So, just to give a quick overview of what I'm going to be talking about today, I just want to start with some background information. I know you guys have been hammered with background information on aspen and molecular tools to this point, so I'll just quickly go over things that are especially pertinent to my study. I want to give some specific background on aspen, just introduce you guys to the term phylogeography, and briefly explain what a phylogeography is, as that's the basis of my project. And then I'll give an intro to sources of genetic variance and how genetic variance is shaped across the landscape. Then just briefly discuss the specific goals of my project and the methodology we used. And then get to the good stuff and go over the results we have to this point, and then just wrap everything up with a summary. So just to give more background information on aspen, a lot of this has already been covered to this point, but I might mainly want to focus in on aspen's distribution. And aspen has an enormous distribution. It's the largest natural distribution of any tree native to North America. It stretches all the way from Labrador in northeast Canada across the entire continent into Alaska, and it also stretches south through the western United States all the way to mid-Mexico. An interesting fact about aspen's current range is that most of it was formerly covered by ice during the Pleistocene and the last glacial maximum, which means that aspen's undergone a significant and geologically recent range expansion. And finally, as you already know, aspen's capable of both sexual and asexual reproduction. And sexually, aspen reproduces through both seed and pollen, and these are adapted for long-distance wind dispersal, and this allows for long-distance gene flow. And asexually, by propagating through the root suckers that have already been discussed today. Okay, so here's phylogeography. What a phylogeography actually is, it just deals with spatial arrangements of genetic lineages within a species. So it allows you to actually link landscape features with patterns of gene flow. And when you're trying to put patterns of gene flow and genetic variance on a map, some sources of genetic variance are things like mutation, gene flow, genetic drift, and selection. And those things are shaped first by landscape features such as mountain ranges and basins, prevailing winds, soil conditions, and competition with other species, as well as historical processes like glaciation events, range expansion and contraction, and population size fluctuations. So here's our map showing the current range of aspen. And as I said, it's enormous. It stretches the entire continent, and it's the largest of any tree native to North America. So the main objective of my thesis project was basically just to characterize the distribution of genetic diversity across the range and to assess any population structuring that may be occurring on the range. So we had two hypotheses going in. One is that aspen expanded its range from southern glacial margins as well as northern glacial refugia. And what you'd expect to see in this case is increased genetic diversity in these populations from the southern glacial margins, as well as populations that represent putative refugia. And our second hypothesis, which is not mutually exclusive from the first, is that aspen has maintained a stable southern edge throughout multiple climate oscillations. And again, similar to the first hypothesis, what you'd expect to see in this case is increased genetic diversity in these populations in the southern portion of the range. An alternative to hypothesis 2 is rather than a stable edge, you could have a trailing edge as the range expands northward. And in this case, you'd expect to see genetic diversity similar to that in the rest of the range, or even decreased as those populations may be becoming isolated. So here's a map showing aspen's range again in green. and outlined in blue is the last glacial maximum, the extent of the Laurentide and Cordilleran ice sheets during the Pleistocene. And this just illustrates what I said before. As you can see, the vast majority of Aspen's current range was under ice during the last glacial maximum. And so it's already been shown for several other taxa that glacial refugia served as points of recolonization. So we wanted to assess that with Aspen. So to do that, we have a sample set representing the full extent of the current range. We have sample sites representing putative refugia, so we could test our hypotheses. Mainly, we have populations in northern Alaska, where the Beringia refugium is, or was, and the Great Lakes area, where there's also a glacial refugium there. And then we also have the western United States sampled extensively, as those populations lie south of the last glacial maximum, to assess our stable versus trailing edge hypothesis. And the molecular tools we use, these have already been introduced, so I'll just go over this really quickly. Microsatellites are stretches of DNA that consist of short tandem repeats. The example that I have here is just CTA repeated X number of times, a trinucleotide repeat. The most useful thing about microsatellites is they're highly polymorphic, as Karen already explained, highly variable, which makes them an appropriate tool for genotyping individuals. And for this project, we used 8 different microsatellites. And the statistical method we use to assess structuring across the landscape is a software program called Structure. And Structure just employs a Bayesian algorithm that allows you to identify groups of genetically homogeneous individuals based on a multi-locus data set. And it just calculates a probability of individuals and populations belonging to any number of clusters. And the number of clusters to be tested is determined a priori by the user. So you can test any number of clusters across the landscape and have Structure test all of them. And then there's a statistical method you can use to identify the most likely number of clusters on the landscape. I won't go into detail on that. This is easiest to explain by just showing an output from Structure. So this is the result of our first. We ran all 30 of our populations, and we tested 1 through 10 clusters. And that using that statistical method I just mentioned, Structure identified 2 clusters on the landscape as the most likely number of clusters. And those are represented here by the 2 colors. You have a blue cluster and an orange cluster. And the populations, the 30 populations, are labeled along the bottom of this bar graph. And within each population, there's an individual bar which represents an individual in that population. And the color of the bar represents the probability of that individual aligning with either one of the 2 clusters identified by Structure. So hopefully that makes sense. And the interesting finding here is our first cluster identified by Structure, this blue group, represents all of our populations from the western United States, the southwestern portion of Aspen's range. We have Arizona, Idaho, the Lake Tahoe area, Oregon. We have a Mexico population included, New Mexico, Nevada, Swan Flats, Utah, and a Washington population. And this is easier to visualize on a map. So along the bottom here, we just have the same Structure bar plot that you just saw on the previous slide. And we have our first cluster, the blue cluster. Those populations are color-coded blue. And then the orange populations represent the entire rest of the range. So the next thing that we wanted to do is look for any subdivision within these 2 clusters. So we separated them out and ran a structure analysis on each group to see if there was any further structuring within them. And when we did this for our western cluster, that blue group from the previous slide, we found once again that k equals 2 was the most likely number of clusters. And there's subdivision where we have our southernmost populations grouped together nicely, and then a group that's almost a west coast northwestern group with Idaho, Lake Tahoe, and Oregon and Washington. Maybe put it on a map because it's easier to look at on a map. That's the subdivision that we found within that western group. And I'm not sure what to make about this result at this point, but at some point I would assume that there was some sort of barrier between these two groups that isolated them from each other, and they diverged. But when we did it for the next group, the orange group from our first run, once again, 2 was identified as the most likely, but as you can see, there's no real structuring going on here. All of these populations and individuals within populations have almost an equal probability of aligning with either one of the 2 clusters identified, so there's no real structuring going on in that group. So the next thing we wanted to do was, based on our structure results, we wanted to look at genetic diversity between groups. So we measured allelic richness for each population, and that's shown in this figure on the top, and it's color-coded to match that our structure results. That blue group is our western group, Western United States group, and orange is the rest of the range. And there's significantly less genetic diversity in our western populations versus the rest of the range. And when we pull those together and take an average allelic richness value for each cluster, again, it's significantly lower in the western group. And lends pretty strong support to our trailing edge hypothesis rather than a stable edge during multiple climate oscillations. These populations likely became more isolated over time and therefore less diverse than the rest of the range. It's more connected. Yeah.
If that's a sort of an interpolation map, how does the singularity of that one point affect how the land, we call it landscape, but the continent is affected?
Yeah.
That
You can adjust the settings in ArcGIS based on how many points in proximity, how many of your input points are factored into creating the surface around them. But it would definitely be worthwhile to sample that area more extensively just to try to fill in the gaps. I mean, for instance, the area that's shown yellow between Alberta and Alaska, I would think if we sampled that more extensively, we'd probably have, more red, higher diversity in that area. I don't think in reality that that's what's going on, that there's a gap of lower diversity between Alberta and Alaska. But yeah.
Sort of similar vein, I was interested in that outlier in Mexico.
Yeah.
In any of your analysis, did it separate out from those in the Four Corners states?
It never separated out on its own, and it always, when we looked for subdivision within the Western group, it always strongly aligned with the rest of the southern United States populations, like Arizona and New Mexico. But I'm not sure what to make of that, but I would say that the fact that Mexico has by far the lowest genetic diversity of any of the populations we have, I would say that as the range shifted northward, those Mexico groups were left behind a long time ago and have become increasingly isolated. And so they're definitely suffering from lower genetic diversity than the rest of the range. And I think that trend will continue, definitely. Yeah. Yeah. So that's one that has us a little bit perplexed. I really expected the Yellowstone population to not only cluster with the western group, which it didn't. It was one of those orange populations that clustered with the rest of the range. But I also expected it to have genetic diversity similar to that in the western group, maybe be a little less. The only thing I can say about that at this point is most of the samples that we included from that group came after fire or after a disturbance. So I don't know if there's more sexual reproduction there, which would account for, higher diversity in that group. But there's something strange there. I have to think about that.
There's Lupin. We have a couple of minutes left.
And with that, I'd just like to acknowledge my advisor, Karen Mock, my committee members, Paul Wolf and Jim Long, as well as everyone who helped me along the way, and especially the money that made it all possible. Yeah.
Question, the last map you showed, I wonder how much that lone point would